Okay, so we have made it kind of to the end. Um, if we take a look back, we have looked at a lot of processes that happen inside of a cell. We have looked at signaling, we've looked at transport in and out of a cell, we've looked at transport inside of a cell, vesicular transport. We've looked at some of the biochemical things that occur in cell with the generation of energy, photosynthesis. We've looked at the different organelles and what they do. We took a pretty significant look at the plasma membrane. So we've looked at all of the components and pieces of the cell. The last thing that we're going to do here is we're going to talk about how cells interact with each other and sort of form communities of cells. So cells are generally going to be organized into tissues. If we're talking about a multicellular organism, then we're going to see that there's going to have to be some type of organization into some type of tissue. Humans have 4 types of tissues. We've got nervous tissue, muscle tissue, epithelial tissue, and connective tissue. Now, when we think about sort of comparing plant and animal cells, we can think that there is a very different overall function of these cells. Animal cells generally need to be able to move. So in general, animals have to go find their food, and they could possibly be preyed upon as someone else's food. So they're going to need to be able to be very strong, very agile, have the ability to move very rapidly, and force is going to need to be able to be transmitted through whatever cell community, whatever tissue, very efficiently. Plants, on the other hand, tend not to move. So their cell and the structure and the tissues are going to require a lot more rigidity. So they need to have a tissue structure that is going to support their overall structure, which needs to be upright. Okay, so depending on the type of cells that we're looking at, we're going to see different functions in these tissues. So we've already looked inside of the cells. We know that there is some structural components inside using the cytoskeleton. Now we are going to look outside. We're going to take a look at 2 different things that are going to sort of bring cells together to form these communities or these tissues. We're going to take a look at the extracellular matrix, and then in our next lecture, we're going to take a look at cell junctions that are going to help hold cells together. So we're going to start with plants. We're going to take a look at the extracellular matrix in plants, which is going to consist of these cell wall structures. So plants synthesize, secrete, and completely control the composition of their cell walls. We tend to see that as a cell is growing, the cell walls tend to be a little bit thinner. They're called primary cell walls, and they actually allow for the growth of the cells. Once the cells have actually reached their growth potential, then we tend to see them lay down what are called secondary cell walls that are a lot thicker and have a lot more structure to them. So cell walls are made of polysaccharides, particularly cellulose, which you can see here in blue, and pectin, which you can see here in green, are the 2 major components. Now, cell walls are going to provide structure and provide some protection and shape for the cell to kind of have a ability to either push up really close onto that cell wall or draw back from it. Generally speaking, plant cells want to be pushed right up against the cell wall. And to do that, they need to have enough water in them. So water is going to provide in the plants what's called turgor pressure. So here what we're looking at is a cross-section through a leaf of lettuce. And this is a very crisp leaf of lettuce. And so you can see that there's no wilting in here. That means that the cell membranes are pushed way up against the cell walls, and it's essentially pushing out on the cell walls, and it's providing that nice, crisp, rigid structure that is going to be associated with a healthy plant. As the plant would begin to lose water, the cell membrane would pull away from the cell wall, and that's when we would begin to see wilting, which is not good under any circumstances. So we've all seen this with a house plant. It begins to wilt, it loses its structure, you give it some water, stands right back up. The difference there is how much the cell membrane is being pushed up onto the cell wall in the overall plant. So let's take a look at cellulose. So we'll use that as our major example here for the plant ECM. So we have, starting down here with the plasma membrane. So this would be the plasma membrane of the plant cell. And then you have the cell wall structure, which the primary component is going to be cellulose. Cellulose is these green bars here that you can see are laid down in kind of a almost like a grid shape. Cellulose is the most abundant organic macromolecule on this planet because of its concentration in plants. This is where plants get their tensile strength. This is where they're going to be able to resist a tearing, if you will. Now, the cellulose molecules are going to have to be linked together, and we're going to link them using glycans, which are these red fibers here. So this is how we sort of hold all of this together, is to cross-link using the glycans. We also have pectin, which are the blue guys in here, okay, that are basically going to provide some resistance to compression. So we don't want to be able to compress the cell wall, and so the pectin component is going to give us a resistance to compression. The yellow layer, called the middle lamina, is what cements the neighboring cells together. So on the other side of this middle lamina here would be the next cell wall, and then the membrane up here. So this is sort of the glue that would hold, I'm going to go back to this picture, this is the glue that would be holding these cells together here. So cellulose is going to have to be synthesized by the plant. If the plant controls the composition of the cell wall, then we're going to have a way to synthesize this. So we have an enzyme called cellulose synthase, very logically named, that is going to synthesize cellulose in a plant cell. So most of what we've talked about so far regarding anything that a cell is making has been made inside the cell and then released via exocytosis. Cellulose is different. We have the cellulose synthase complex, which you can see right here. And what it does is it's completely embedded in the membrane here. So it's embedded in the membrane. And instead of us making the cellulose down inside the cell and then moving it out, what's happening is we are using this complex to move the monosaccharides and the disaccharides out that then are added to this pre-existing cell wall structure. So essentially, you're constantly adding these cellulose microfibrils into the cell wall by pushing the subunits through this enzyme. So the actual cellulose is never actually made inside the cell in the cytosol. It's being made as it moves out of the plasma membrane. Now, this entire membrane complex actually moves, so it laterally diffuses within the membrane. So it's constantly moving. So you can see this arrow here saying that this would be moving along in this direction. They actually move along microtubules that lie beneath the plasma membrane. The enzyme complexes are connected to the microtubules, so the cytoskeleton, using connector proteins. So basically, the arrangement of the microtubules in the cytoskeleton is going to dictate the arrangement of the cellulose microfibrils in the cell wall, because these enzyme complexes can only move down the microtubules. So there's a direct connection, really, between the cytoskeleton, the internal structure, and the extracellular matrix, the external structure. So now we're going to move on to animals. So in an animal cell, our major extracellular matrix protein is going to be collagen. Collagen is providing tensile strength to the animal tissues. So depending on what type of tissue we're looking at, we can have lots of different types of collagen. So mammals have at least 20 different genes for collagen. So different types for different things. A collagen molecule right here is actually 3 collagen polypeptide chains sort of wrapped around each other. So this is a collagen molecule. They aggregate to form collagen fibrils, which then aggregate to form collagen fibers. Collagen fibers you can see running through a tissue here. So here the fiber is running in this direction, and here the fiber is running towards you. So you're looking at a cross section through this. So there is going to be an arrangement of the collagen fibers that's going to provide tensile strength in the directions of the fibers. So this tissue that we're looking at here would have tensile strength in both this direction and the direction coming out at you here. So the thing with these collagen fibrils is that they will spontaneously form when you have these collagen molecules coming together. So the collagen molecules spontaneously aggregate to form the fibrils. We actually don't want that to happen in the cell. So unlike the cellulose we just looked at, these collagen molecules are actually made inside of the cell and have to be secreted via exocytosis. We don't want them to aggregate and form fibrils, which can then aggregate to form fibers inside of the cell. So we're going to have to secrete them in an inactive form. So what we have is procollagen. So procollagen is going to have these sort of inactivating domains on either end, and that's what's actually going to be secreted into the extracellular matrix. Then we've got an enzyme, a proteinase, that's going to come along and is going to chop off these inactive green portions to give us an actual collagen molecule that will then self-assemble into a fibril, which self-assembles then into a fiber. So this is a way for us to synthesize this this molecule, but make sure that it doesn't begin its aggregation until it leaves the cell. If it did aggregate in the cell, it would essentially choke off the cell. There would be so much collagen in there that nothing else would be able to do anything. Now, this guy right here, his name's James Morris, and he was known as the elastic skin man. So you can see his skin was very, very stretchy. Essentially, he had no tensile strength, which comes from collagen. Many times disorders that would leave the skin like this are actually due to genetic conditions where the proteinase does not work. So if you secrete procollagen, but it can never be sort of activated into collagen, then you never get your collagen fibrils, you never get your collagen fibers. So there is a benefit certainly to secreting it in an inactive pro-collagen form, but only if you actually have the enzyme to activate it. So one of the things is that collagen definitely has to be arranged in the correct direction in order to function properly here. This is the arrangement in skin, and you can see that it's going in multiple directions, and so we have tensile strength in multiple directions. The cells that are going to secrete this collagen, which are fibroblasts, are also going to help arrange it. So this is a still from a video that you're going to be able to see here, where the fibroblasts actually they crawl over the collagen and actually sort of pull it in the direction that we want it to go. Just making the collagen is not enough. We have to make sure that it is arranged properly as well. This is really important for things like wound repair, so that we make sure that we can align the fibers properly so that the tissue does function again. So just like we saw with our cellulose, there is definitely a connection between the extracellular and the cytoskeletal elements. So in the cellulose, we saw that the enzyme making the cellulose is connected to the microtubules in the cytoskeleton. In animal cells, we also have a connection via a protein called fibronectin. So fibronectin is kind of this almost like a pincher or a kind of a Y-shaped molecule that is going to both grab a hold of the collagen and a cellular protein. So if you look at this, here's our collagen and here's our fibronectin kind of grabbing onto it like a pincher. Now, the fibronectin is not going to be directly attached to the plasma membrane. We're going to need another set of proteins called integrins. Integrins are actually membrane spanning. Okay, so remember we would have a hydrophobic region in here that would span the membrane and hold the integrins in place. The integrins are going to bind to these cell attachment spots on the fibronectin, and then in their intracellular domain, they're going to bind to adapter proteins which bind to actin. So here you have a connection between actin, the cytoskeleton, and collagen, the extracellular matrix. Now, integrins are specific for given cells, and problems with them can lead to different diseases. Several different immune disorders can be traced to integrins. So we've got something called leukocyte adhesion deficiency, where your leukocytes are incapable of adhering to what they need to adhere to because of an integrin issue. We've got blood cell disorders where our blood platelets can't bind to the necessary tissues, so we end up having a bleeding disorder. Okay, we have actually talked about integrins before when we talked about how fibroblasts move, so this should look familiar to you. And what we were talking about is actin. So here's our actin, and here's our integrins, and this is the extracellular matrix here. So when a fibroblast moves, so this yellow here would be our substratum, which in this case would actually be the extracellular matrix. It's the integrins that actually interact with this extracellular matrix and then allow the actin to polymerize and change shape. So what we've talked about before, which is the crawling of a fibroblast, relies, again, on this connection between the cytoskeleton and the extracellular matrix. So this was from a previous chapter. You should sort of remember it, hopefully. Another portion of the extracellular matrix has to do kind of with space filling and resisting compression and kind of forming gels and things like that. So GAGs are glycosaminoglycans, which is why we call them GAGs, because that's a huge word. They're negatively charged polysaccharide chains that are made of repeating disaccharide units. So you just have the same units over and over and over, and then they are linked to these core proteins via covalent linkages to form what are called proteoglycans. Now, there's all kinds of negative charges here, and negative charges are obviously going to draw in positive charges. Particularly, we're going to see that these draw in sodium. Sodium then obviously draws in water. So these proteoglycans draw water and form gels. Some of them tend to be very, very gel-like. Okay, and they might be used to fill space, like the posterior chamber of your eye. Others tend to have more collagen in them, and so it's a much heavier, denser tissue, and that would be something like the overall matrix in the cartilage that is holding your knee together, which can withstand huge amounts of force. So there's definitely variation within this, within the proteoglycans, and exactly how much water, whether they're being utilized more for strength or they're being utilized to just fill up a space. But they all have at least some draw on water because of the draw on sodium. And these are going to help provide either that structure or space filling function to the extracellular matrix in the animal cells.